Hello, everyone, and welcome to another episode of JAX Live. I'm your host, Jenny Morganwick, and joining me today to talk about our diversity outbred mice is Kelly Brackett and Lizzie Axton. Kelly is a scientific advisor at our Bar Harbor campus. She has a background in evolutionary genetics, and it's actually what she studied for her PhD. And she also manages the Diversity Outbred Colony, where she provides oversight on how we're conducting the breeding. She provides scientific advisement. And Lizzie is a technical information scientist out of our Sacramento campus. And she's actually been working closely with the researchers who developed the diversity outbred mice to generate resources and educate the community on how to use these mice and what they can be, how they're valuable for scientific research. And she's also our subject matter expert from TIS on these guys. And she also did her PhD work in pharmacology and toxicology. So she's a great person to talk with us about how these mice can actually be used in research. So how are you both doing today? Welcome.
Doing good. Great. Thank you.
Thanks for joining us. So before we get into our conversation about these diversity outbred mice, I did want to let the audience know that they can participate in the interview today. They can ask questions. They can use the comment box on whichever platform that you're joining us on and ask your questions, give us a shout out, let us know where you're tuning in from. I love to hear where people are tuning in from. Usually we get people joining us from all over the world, so it's nice to see that. And we'll take any questions that you have. We'll pepper them throughout the interview today. And if we get a bunch of them, we can include them in our live Q&A session at the end of the interview. So make sure you stay tuned for that. So Lizzie, Kelly, let's dive right in. So we all recognize that the human population is incredibly diverse. And there's an infinite number of genomic variants that can determine both a predisposition to disease and also the response to treatments. So, I don't think anyone would argue that it's important to have a mouse model option that encompasses that level of diversity. So for our audience today, Kelly, can you give a general overview of the diversity outbred mice? Because, they are relatively new in the grand scheme of science, and I'm not sure everyone in our audience may be familiar with them.
Sure, I'd be happy to do so. So the JAX diversity outbred, or as we call them, the DO, have a random assortment of genetic background from 8 different inbred strains. And this eventually results in a population of mice that more closely mimic the genetic diversity observed in human populations. The 8 founder strains were chosen specifically because of their unique characteristics. For example, one develops type 1 diabetes, that's our NOD strain. Another is prone to cancer, that's AJ. And a third model develops obesity, that's the New Zealand obese inbred strain. And that's just 3 out of the 8. Each founder has also been sequenced, their whole genome, so we know where every SNP, single nucleotide polymorphism, is in their genomes and the differences between them. First, the 8 founder strains were crossed in a precise breeding scheme to generate the collaborative cross. The CC are a series of recombinant inbred lines, meaning that each line is a unique mixture of the 8 different founder strains, but within each line, the mice are genetically identical. So you can actually repeat experiments with each of these lines. And these lines are also available at the Jackson Laboratory if anybody's interested. Then the CC lines were then randomly crossed together to generate the first generation of the diversity outbred. Since then, we've been maintaining this population by random breeding for 40 generations. And each mouse in this population is genetically unique and cannot be replicated. It's just like humans, each of us are unique. By mixing the genetic backgrounds of these 8 inbred strains, we basically generate a diverse population of mice that can be used to model either susceptibility or resistance to a wide range of human diseases and conditions.
So you're saying that researchers can use these mice to get a better idea of disease susceptibility or severity or the resistance within a population. So have these mice been used already to study a variety of disease areas, or has the field been focused on one disease in particular?
Definitely the variety. So this population has been around since 2012, and they've been used for research applications like pharmacology, toxicology, oncology, type 2 diabetes research, infectious diseases, and behavioral research, to name a few. So, anything is sort of game with this population. And because of the inherent diversity in these mice, researchers can basically explore a bunch of discovery-based questions like the genetic, environmental, or gene by environment basis of disease using high-resolution genotyping by use of the SNP data and quantitative trait loci mapping. They can also use the population as a proxy for the human population in assessing a variety of biological responses to disease treatments like lifestyle changes, pharmaceutical compounds, or even vaccines. And researchers continually surprise us with the new and creative ways that they choose to use this population in their research.
So, knowing that these mice have been used in multiple areas, this goes into my next question. And Lizzie, I'm going to shoot this over one to you, since you're actively developing the resources for this diversity outbred line. So, I've heard of the diversity outbred database that is hosted by the Jackson Laboratory. Can you tell us a little bit more about that database and how a researcher might use it?
Yeah, I'd be happy to. So the Diversity Outbred Database, or DODB, is just one of the many resources that has been developed as part of the JAX Genetic Diversity Initiative, or JEDI. And I find the DODB to be particularly useful because it is a collaborative database where researchers can either download data sets that others have produced and can also upload their own data sets for sharing. And there's all kinds of helpful information in this database, including the genotyping results from the SNPs that Kelly mentioned, and then also just phenotyping results, including things like omics data sets. And how a researcher might want to use this is if you're new to DO mice, what you could do is look and see if there's any data sets from within your research field to see if you could perhaps download those data sets and sort of reassess them to ask a different research question and potentially use this for like preliminary data for a grant or something like that. It could also be a helpful way to learn just what types of data are produced from DO data sets, and also even practice data analysis for things like QTL mapping if you're new to that field. And overall, I would say if you're working with DO mice, definitely go ahead and upload your data sets so that everyone else can utilize them as well.
So is this database, is it free to access and use? How does someone gain access to this?
Yes, so this database is free, as well as basically any of the other information that's on the Genetic Diversity Initiative page. Probably the best way to get there is to go to the strain data sheet for the diversity outbred mice, the JADOs, and right at the top there's a link that will take you to the JEDI resource. And really beyond the DODB, there's some background information on DO and other mouse models of diversity, literature so you can see how others have used them, guides for data analysis, really anything that you need to get started in working with these mouse models of diversity.
Oh, that's excellent. So we're getting a couple shout outs. So we've got some people from Boston, we've got someone from Maine, California, and we've got someone saying hi from India. We've got New Jersey. So we're getting a lot of people tuning in, and we actually we actually do have a question from someone in the audience. Someone is asking, how are these different than outbred mice like CD1? And my follow-up question to that, because I was thinking the same thing, is there an advantage of using the DO mice over other outbreds? So maybe Lizzie, you can take that one.
Yeah, I'd be happy to. So As many of you are probably aware, outbred mice in general are not new to the mouse laboratory. They've been around really since the start of mouse models. And they are a really useful tool. And I would say primarily they've been used for applications like toxicology. They have a couple of limitations that you should be aware of. The first of these is that these are produced by breeding. Basically, instead of inbreeding with brother and sister matings, you are setting them up as non-brother and sister matings, and this helps promote genetic diversity within a population. However, I would say this results in a pretty low level of genetic diversity, so some of these don't have much more diversity than what you would see in an inbred population. The second thing is that since these are essentially randomly bred, you can have genetic drift over time, and you can end up with subpopulations of mice if you're not careful. And so this sort of impacts the reproducibility element, because if you were to do a study in CD1, say from mice 10 or 20 years ago, they might have different genetics now, and so it might be hard to translate those two data sets. These limitations were some of the original, I guess, motivation for the DO strain. So the diversity outbred mice were developed from the 8 founder strains, and this resulted in a high level of genetic diversity. And this population now has about 40 million SNP variants, which is about half of what you see in a human population. The other benefit is that we are carefully breeding these mice, and so this helps maintain the genetic diversity over time. And actually, my understanding is that's a big part of Kelly's job. So Kelly, did you want to briefly comment on that part of it?
Yeah, sure. I can just say that we carefully examine our random breeding schemes for each generation. We have many checks and balances to make sure that we're not doing brother-sister mating. And essentially, we're ensuring the maintenance of genetic diversity as best as possible.
Perfect. Thanks, Kelly. I would say to add to that too, one of the big applications for DO mice is in mapping genes and phenotypes so that you can look at things like disease progression and severity. And the reason why this is possible with DO is that all of the founder strains have fully sequenced genomes. And so it's relatively straightforward to reconstruct the genome of each unique DO mouse. And so when you do phenotyping, say looking at disease severity, you can sort of map the genetic component to the phenotype that you're seeing so that you can look at these genotype-phenotype associations. And this really isn't possible with a standard CD1 outbred strain.
And that's great, because I know that there's a lot of diseases out there that, you know, we know that the disease exists because of a mutation, say, or something like that, but the severities are all over the place. So, you know, it really exists on a continuum for the population. So these mice are a great way to look at modifiers, it sounds like, too, you know, other genes that could, you know, exacerbate a disease or cause a decreased, you know, severity of it. So this sounds like a great tool in looking at human disease for sure. So Kelly, since you are basically the one that's kind of in charge of all the breeding and the colony and all of that stuff, for people that are interested in working with these mice, can you speak to their availability? Because I know that they're a little bit different than our other inbred strains, and it can be quite surprising to people when they try to get these mice.
Yeah, so the DO do have a unique breeding scheme. And therefore, unlike our inbreds, like you said, they are not offered on demand per se. Essentially, we breed 175 pairs in what we call waves, basically on a quarterly basis. They all get bred simultaneously and they have litter simultaneously. And this is done 4 times a year, so they're offered on a quarterly basis. So therefore, you sort of have to plan in advance a little bit and request your numbers and sex of the mice that you want ahead of time. And we'll basically put it in the queue and fulfill that during the next wave. Additionally, due to the size of the colony, we do have some limitations on how many mice a researcher can order at once. And for more information on the timing of these breeding waves and some of our ordering parameters, I suggest people visit the strain data sheet on the DO on our website, and contact us with any questions that you have specifically to your order or before writing your next big grant that you would like to include the JADO in.
And, you know, thinking about this model, and I know they came around in 2012. So in the grand scheme of things, like I mentioned before with science, it's relatively new. So where do you guys see, and I'd love to hear from both of you on this, where do you guys see the future of this type of model going?
Kelly?
Oh, you don't want to play rock, paper, scissors for this one?
No, you first.
So I really think the JADO and diversity populations in general are sort of just getting started. With this new era of precision genomics and researchers just beginning to understand the need to study diversity, like you said, Jenny, that we don't all respond to therapies the same way. We also don't develop diseases in the same way. That there is a need for this type of diverse population. And I think as researchers find, their preliminary answers in inbred strains, they'll then look to take the next step to diverse populations and see if the phenomena that they're finding are consistent across genetic backgrounds, or if they are inconsistent, why, and finding the genetic basis for that. And they may want to do this before moving to things like human clinical trials with therapies and things like that. Additionally, like I said, I think the DO is the first population of its kind, but it won't be the last. There was a recent, in the past 10 years, a push by the NIH and major publishers to have researchers include both sexes in animal studies. I think we're coming up, and we're not far away from a call from those types of institutions to include diversity in animal studies as well, and the DO would be a part of that. So I predict that over the next 10 years, we'll probably have to grow this JADO population. And there potentially will be new populations that come online, perhaps with different founders that start the population off. And this will all be to meet the sort of emergent need for studies of diversity in mice.
Yeah, I would agree with that, Kelly, and say that I really think that we're barely scratching the surface of what we can do with DO. And I do hope that we'll start to see new populations of diversity models as well. I see two major fields where I think there's going to be a lot of development in the use of DO in the upcoming years. The first of these is in toxicology. Maybe this is my personal bias because that's my background, but the need I see here is that if you were to do a toxicology study in an inbred strain, you're really only looking at a narrow subset of genetics. And so if you were to expand your tox studies into something like DO, where you have a wide spectrum of genetics, I think you're more likely to pick up on any adverse effects from therapeutics, and hopefully make it so that when these drugs make it onto market, we're not as surprised by any of the adverse effects that happen in humans. So I think that this can be a much more translatable model for toxicology studies. The second field I see in, which is probably more challenging, is in model generation in DO. I think if we can add modifier genes to change the susceptibility, for example, make the population more prone to Alzheimer's disease or different types of cancers, to really expand the types of diseases that we can look at with DO mice. And the real benefit here would be that for complex diseases like Alzheimer's, you can look at modifier genes and sort of look at the mouse as a whole in both what's causing the Alzheimer's and what is sort of leading to resistance or susceptibility. So I think that's a lofty goal, but I do think that once we have that possibility, I think we're going to see a lot of growth in the DO population.
Well, it sounds like that there's a bright future for these mice and other lines that come off of them. We're winding down on our time today, so I wanted to thank you both for joining us. It was great to hear from the person, Kelly, who is basically managing this colony and making sure it stays diverse, and also providing that scientific expertise. And Lizzie, it's so great to hear that not only do we have this model, but we also have a lot of resources to back it up that people can access to really kind of set them up for success to use the mice. So I encourage everyone that's listening to check out the strain data sheet for these DOs and access some of those resources to see if they could be helpful in your research. So we really appreciate you guys joining us. If we didn't get to answer your question in the audience, we'll be sure to follow up with you. Next week starts our 3-week humanized mouse live stream series. So that's very exciting. Lots of humanized mice topics to talk about. And that will be with our very own Jim Keck, all 3 episodes. So if you're interested in that topic, make sure you join us, same time, same place. And we will see you all at the next JAX Live. So thank you.
Thank you, everyone.
Thanks, Jenny.